morning. The bibliographic references and the publisher's source article are accessible in the description under the video. If you want to support the dissemination of scientific knowledge, I invite you to subscribe to this YouTube channel. It's free. Also, please, can you click on the thumb pointing towards up? Abstract. Sponges are frequently used in kitchens and have been shown to harbour large numbers of bacteria, occasionally also pathogens. Less is known about kitchen brushes regarding usage and presence of bacteria. In the present study, the use of sponges and brushes was studied in a survey among 9,966 European consumers in 10 countries, and growth and survival of bacteria in sponges and brushes were examined in laboratory experiments. Sponges were the preferred hand-cleaning utensils for washing up in the majority of countries, while brushes were most frequently used in Denmark and Norway. Consumers mostly change their sponges at regular times, but also sensory cues looks dirty, smelly, slimy, and usage occurrences such as wiping up meat juices may trigger replacement. Besides cleaning the dishes, over a quarter of the dish brush users also use it to clean a chopping board off the soilage from chicken meat juices. The water uptake and drying rate varied considerably, both between different sponges and between brushes and sponges, where brushes dried fastest. Campylobacter survived 1 day in all sponges and Salmonella more than 7 days in 2 of 3 types of sponges. In the type of sponge that dried slowest, Salmonella grew on the first day and was always found in higher levels than in the other sponges. Non-pathogenic bacteria grew in the sponges and reached levels around 9 log CFU/sponge. In brushes all types of bacteria died over time. Campylobacter and Salmonella were reduced by more than 2.5 log to below the detection limit after 1 and 3 days, respectively. Bacteriota studies revealed a tendency for a dominance by gram-negative bacteria and a shift to high relative prevalence of Pseudomonas over time in sponges. Both enumeration by agar plating and bacteriota analysis confirmed that the pathogens were in a minority compared to the other bacteria. Treatments of sponges and brushes with chlorine, boiling or in the dishwasher were effective to reduce Salmonella. We conclude that brushes are more hygienic than sponges and that their use should be encouraged. Contaminated sponges or brushes should be replaced or cleaned when they may have been in contact with pathogenic microorganisms, e.g., used on raw food spills. Cleaning of sponges and brushes with chlorine, oiling or dishwasher may be a safe alternative to replacing them with new ones. 1. Introduction. Kitchen sponges are commonly used by consumers for washing up and scouring of pans and casseroles, but are also used for cleaning kitchen surfaces, such as sinks, refrigerators and stovetops. Brushes are the dominant utensils for manual cleaning of dishes washing up in Norway. In an observational study in UK, several consumers used more than one type of cleaning utensil for washing up. 29%, 50% and 77% of consumers used brushes, sponges and cloths, respectively. There is lacking information about the use of brushes in other countries. Sponges collected from consumers can contain high bacterial numbers, in the range of 6-9 log CFU. A wide diversity of non-pathogenic and opportunistic pathogenic bacteria as well as viruses, archaea and eukaryota have been found in used sponges. Presence of the pathogenic bacteria Salmonella and Campylobacter have also been reported. While sponges are used to remove food soils and reduce bacterial numbers, it may also transfer bacteria, including pathogens, to surfaces. Thus, sponges have the potential to act as a reservoir and spread bacteria. There is generally much less information about kitchen brushes and sponges in the scientific literature, and therefore it is unclear to which degree they can act as reservoirs for pathogens. In a study among elderly in UK, total counts in the range 3-8 log CFU were found in used brushes, and Enterobacteriaceae and Staphylococcus aureus were frequently detected. Sponges absorb large volumes of water/fluids. Sponges often remain wet after use, creating a potential for bacterial growth, while rapid drying may reduce the growth or kill the bacteria. Salmonella has been observed to die off in dry sponges, but may grow in humid kitchen cloths. There is limited information in the scientific literature linking parameters, such as water uptake and drying ability of sponges, to growth and survival of pathogenic bacteria. Brushes will likely have less water uptake than sponges, but more knowledge is needed to reveal how this difference affects bacterial growth and survival. Cleaning or disinfection of cleaning utensils may be a way to control the bacterial contamination and limit further spreading in the kitchen environment and will also lead to reduced waste as the utensils will have a longer lifetime. There are several studies on cleaning and disinfection of sponges, but to our knowledge no studies on kitchen brushes. Addition of hypochlorite or dishwashing detergents, the sponges, have been reported to reduce the bacterial levels, I155 log CFU, but some studies report that the antibacterial effect is quenched by the presence of food soil. Sharma et al. 2009 reported that the use of dishwasher or 1-min treatment in microwave oven reduced the bacterial levels on sponges by 5 log or 6 log, respectively. The bactericidal effect of treating sponges in microwave oven and dishwasher has been confirmed in other studies. These studies on cleaning and disinfection of sponges focus on killing of bacteria and not on whether food soil is removed from the sponges. Remaining food soil could allow regrowth of surviving microorganisms if the sponge is humid. To give risk-reducing advice to consumers, more knowledge is needed about the survival and growth of pathogenic bacteria in kitchen brushes and sponges related to consumers' practices for use and handling of these items. In the present study, we examined usage of sponges and brushes among European consumers and evaluated different types of sponges and brushes for water uptake, drying capability, growth and survival of Salmonella and Campylobacter as well as other kitchen-related bacteria. We also tested the effect of different cleaning methods for these utensils. 2. Materials and methods. 2. 1. Web-based survey. 2. 1. Survey questionnaire. A cross-national survey was conducted in 10 European countries including Denmark, France, Hungary, Germany, Greece, Norway, Portugal, Romania, Spain and the United Kingdom. The survey included modules on hygiene and food handling practices for diverse food categories and socio-demographic questions. The present publication reports specifically on a subselection of questions related to washing up of dishes and cleaning utensils in the kitchen with focus on response alternatives concerning sponges and dish brushes. 2. 2. Consumer recruitment and data collection and analysis. Recruitment was subcontracted to a professional survey provider administering a large consumer panel worldwide. In each country, the population sample consisted of private households, selected by stratified random sampling based on the nomenclature of territorial units for statistics level 2 of the respective country and the education level of the target respondent. The within-country stratum sample sizes were proportional to the corresponding population stratum sizes. In the present paper, responses from a total of 9,966 households across the 10 countries are considered. Respondents consisted of 49.5% males and ranged from 16 to 90 years old, mean of 46. 6 years. A bias towards higher education occurred as an artifact of running the survey online, with 59% of the respondents declaring a university education. With regard to food safety, 4 risk groups were represented in the sampled households: pregnant women, families with children under 6 years of age, diabetics and immunocompromised, and elderly above 65 years of age, wherein 47.1% of the households represented at least one of the 4 risk groups. Frequencies of self-reported practices are reported in terms of percentages overall and per country, gender or age group. Cochrane's Q-test was applied to identify significant differences between treatments at a 95% confidence interval. 2. Laboratory studies of sponges and brushes. 2. 1. Bacterial strains. The bacterial strains used in this study are listed in Table 1. The strains from Norwegian and Portuguese kitchens were isolated during consumer visits within the Safe Consume project. The bacteria were identified by 16's rDNA sequencing, and representatives of the dominant bacterial genera identified among isolates were selected. All strains were maintained in 20% glycerol at -80 degrees Celsius for preparation of cultures for addition cleaning utensils. Frozen stocks of Campylobacter strains were streaked on MCCDA oxide and grown at 37 degrees Celsius under microaerophilic conditions. Campi Jensen 0035A oxide, and thereafter the strains were cultured in 2 steps in Mueller-Hinton broth oxide, incubated at 37 degrees Celsius with 150 rpm agitation overnight under microaerophilic conditions. Salmonella was cultured on tryptone soy agar TSA oxide at 37 degrees Celsius followed by cultivation in 5 ml TSB triptych soy broth, oxide at 30 degrees Celsius with 150 rpm agitation of overnight. Kitchu-related bacteria were cultivated on TSA followed by TSB, as for Salmonella, but at 30 degrees Celsius in all steps. 2. Sponges and brushes. An overview of the characteristics of the 3 sponges and 3 brushes tested is shown in Supplementary Table 3. Photos are shown in Supplementary Fig. S1. 2 of the sponges, S21 and S22, contained the antimicrobials silver and quaternary ammonium compounds, respectively. The brushes and sponges were bought in large retailers in Norway and Portugal and may then be regarded as commonly used in kitchens by Norwegian and Portuguese consumers. 3. Water absorption and drying capacity of new sponges and brushes. The water uptake capacity and water loss during drying of 3 types of new sponges no. 13, 21, 22 and 3 brushes no. 12, 32, 34 were measured. The utensils were weighed before and after being immersed in water 22 plus or minus 1 degrees Celsius for 1 min while being pressed up and down 5 times and allowing excess water to drain off for 1 min. Half of the sponges were squeezed once with one hand after the water uptake and weighted. The drying time of brushes hanging and sponges laid with scrub side up on a clothes horse in contact with air on all sides was determined by weighing after 4, 5 plus 0, 5H and 24H. The temperature in the room was 20-22 degrees Celsius and the humidity, which was influenced by outside weather and varied between the replicates, was in the range 15–40% RH. 4. Survival of bacteria in new sponges and brush over 1 week. 3 types of new unused sponges no. 13, 21, 22 and 1 brush no. 32 were inoculated with a suspension of a mixture of Salmonella, Campylobacter and bacteria isolated from kitchens and a food soil suspension. The bacteria were grown individually overnight, mixed in equal volumes and added to the food soil suspension in a concentration of about 105/ml. The food soil suspension was a mixture of 0.1% poultry soil, 0.1% egg-based soil and 1% lettuce soil, prepared as described previously. The sponges and the brush were inoculated and soiled by immersing them in the bacteria soil suspension 5 times 104 CFU/ml. Sponges were immersed in 200 ml bacteria soil suspension room temperature pressed up and down 5 times while immersed, squeezed once, and placed on a steel tray. Brushes were immersed in 100 ml bacterial soil suspension, pressed up and down 5 times while immersed, and shaken before hanging to dry. To simulate regular use, soil no bacteria was added after 3 and 7 days of storage and water after 1, 2 and 6 days of storage by the procedure described above. The temperature and humidity in the room were registered during the experiment. Samples for microbial analysis were taken after inoculation and 1, 3 and 7 days of storage. Sponges and brushes 3 of each type were transferred to bags. 50–100 ml buffered peptone water was added to each bag, and microbes suspended by stomaching sponge or hand massaging of the bag brush for 6 days. Enumeration of bacteria was performed by spread plating on PCA total variable count, 30 degrees Celsius, 2 days, XLD Salmonella oxide, and MCCDA Campylobacter. Initial tests showed that the enumeration of Salmonella and Campylobacter was difficult due to overgrowth of the plates by non-pathogenic bacteria. To handle this, samples on XLD 1 day and MCCDA 2 days were incubated at 42 degrees Celsius instead of 37 degrees Celsius. Controls confirmed growth of the pathogens at 42 degrees Celsius. The experiment was performed with 3 technical replicates of each type of sponge and the brush. 2. 5. Bacteriotic analyzers. 2. 5. 1. DNA extraction. A volume of 40 ml of stomacher suspension described in Chapter 2. 4 or 5 ml of the bacterial cocktail used for inoculation of sponges and brushes was centrifuged at 13,000 times G for 5 min. DNA was extracted from the pellets using the Kyogen Easy Power Soil HDP96 including beating using fast prep 96 2 times 1600 rpm, 1 min and following the manufacturer's protocol. 2. 2. Bacteriota sequencing and data processing. For all samples, PCR was performed in triplicates and paired-end sequencing 2 times 150 bp was performed using the protocol presented in Caparaso et al. 2012. Briefly, the V4 region of the 16's rRNA gene was amplified with region-specific primers, 806R that included the Illumina flow cell adapter sequences. The forward amplification primer also contained a 12-base barcode sequence that supports pooling of different samples. Samples were purified with Ampere Agincourt Bioscience Corporation and quantified using the quantity picogreen dsDNA with picogreen before pooling. The sample pool was purified and quantified as described above, diluted to 4 nm and sequenced using the MiSeq reagent kit V3 on a MiSeq Illumina following the protocol provided by Illumina. In addition to the experimental samples, the MiSeq run also contained a control library made from PhyX Control V3, which, in this run, accounted for 10% of reads. The library quantification and sequencing were performed at Nofima. The MiSeq control software MSO version used was ART-AV1, 1854. The sequences were processed in KIM2 2019. 1. Briefly, the data was demultiplexed using Demux. Paired ends were joined using vSearch, quality filtered based on a Q-score above 30, denoised using Debler, and taxonomy was achieved using Classify Sklern with the Green Genes 1613-8 database. Sequences originating from mitochondria and chloroplast, very few were filtered out of the final dataset. The taxonomy and feature table was exported to text files and further processed in Excel. The dominating representative sequences were compared to the sequences of the inoculated stains to confirm the identity of the taxa. The sequences representing Enterobacteriaceae were also submitted to Blast Nucleotide Search https://blast and cboi NLM NI gov to get more information about possible genera. 2. 6. Antibacterial activity of sponges. According to labeling, 2 of the sponges S21 and S22 contained antimicrobial compounds silver and quaternary ammonium compounds, respectively, to stop microbial growth and odors. Inhibition of bacterial growth was tested for pieces of sponge 2 times 2 times 0. 5 some in 2 types of diffusion assays. A test where the pieces were placed on an TSA plate where bacteria had been streaked in high levels to form a lawn, and a test where pieces of sponges were embedded into a soft agar 0. 7% containing a bacterial suspension approximately 106 CFU/ml. The target bacteria tested individually were the Staphylococcus, Pseudomonas and the 2 Salmonella strains listed in Table 1. The plates were incubated at 30 degrees Celsius and inspected for inhibition zones. A control known to create inhibition zones, a cloth placemat containing zinc pyrithienidire, Portugal intended to absorb water from wet dishes, was included in the study and tested as described for the sponges. 2. 7. Evaluation of cleaning methods for sponges and brushes. The efficacy of different cleaning methods for eliminating bacteria and reduce regrowth after treatment was evaluated on sponges and brushes inoculated with bacteria and food soils and stored for 1 week. New unused sponges S13 and brushes B32 were inoculated with bacteria suspended in food soils as described in Chapter 2. 4. But using 500 ml suspension, the items were stored humid under plastic film. Added water brushes and sponges immersed in 100 and 200 ml water, respectively after 1, 2 and 6 days. After 3 days, the cleaning utensils were resoiled immersed in 100 and 200 ml food soil for brushes and sponges, respectively. Unsoiled sponges and brushes were included as controls. After 7 days, the cleaning utensils were subjected to different decontamination treatments, as described in Table 2. The treatments were chosen from the literature and observations made during kitchen visits within the Safe Consume project. 3 parallel cleaning utensils were subjected to each treatment. After the treatment, the 3 utensils were handled and analyzed in one of the 3 following ways: 1. Analyzed for total viable count and salmonella directly. 2. Added salmonella about 1000 CFU per cleaning utensil without food soil to test whether soil remaining after the initial treatment could support growth of salmonella. Salmonella culture was washed twice and resuspended in 0.9% NaCl. Sponges and brushes were immersed in 500 ml suspension containing 5 times 104/ml of bacteria and pressed up and down 5 times. Then sponges were rinsed and brushes shaken before being placed in a plastic bag and stored at room temperature for 2 days, followed by analyzers for total viable count and salmonella. 3. Added heat-treated food soil to test if a low number of bacteria below the detection limit remained after the initial treatment and could regrow in the presence of soil. Heat-treated 115 degrees Celsius food-soil mix was added in volumes, as described for treatment No. 2. The cleaning utensil was placed in a plastic bag and stored at room temperature for 2 days, followed by analyzers for total viable count and salmonella. 3. Results and discussion. 3. 1. Use of washing-up utensils among European consumers. The survey conducted on 9,966 households showed that methods for cleaning of dirty dishes vary considerably between European countries. Consumers use preferably a dishwasher, with 44% overall reporting this as their typical method. Fig. 1. Dishwasher usage varies in correlation with household equipment. Pearson R equals 0. 9 of 57% of the households are equipped with the dishwasher, and usage prevalence varies between Norway 69% typical usage for 81% equipped and Germany 59% usage for 72% equipped on top, down to UK 31% usage for 47% equipped. Hungary 24% usage, 30% equipped, and Romania lowest with 10% usage for 15% equipped. If not using a dishwasher, the most frequent practice reported is washing up in a sink or bowl with hot water and detergent, using a sponge. On average for the 10 countries, the use of sponges was 2.5 times higher than the use of dish brushes, 36% against 14% for brushes, but there were large variations between countries. Fig. 1. For example, in Portugal the use of sponges was 6 times more frequent than brushes, whereas in Norway the use of brushes was 3 times more frequent than the use of sponges. Only 8% of the households declare using a cloth as their typical utensil for doing the dishes, with highest prevalence in Romania 19% and lowest in Greece 2%. We also noted that consumers combine cleaning utensils. Thus, of the 36% overall who essentially do the washing up with a sponge, 26% also use a dishwasher, 17% a dish brush and 11% a cloth. Of the 14% overall who essentially do the washing up with a brush, 38% also use a dishwasher, 44% a dish sponge and 16% a cloth. Further, we investigated how dish sponges and brushes are used among their most frequent users. An analysis focused on dish sponge users specifically, N equals 3,578, shows that 56% also use this utensil for cleaning the kitchen countertop. This behavior is especially salient in France, 74% of the dish sponge users, and weakest in Norway, 30%. However, sponges are less utilized for cleaning hazardous soilage such as chicken juices on the countertop, 26% of all dish sponge users, or on a chopping board, 27% of all dish sponge users. In this case, consumers often reported using alternatives, such as a cloth or kitchen roll. Further, in between usage occasions, sponges are mostly kept beside the sink, 62%, followed by hanging 16%, left in sink 10% and left on counter 7%. Hungarian and French consumers most typically reported leaving the sponge beside the sink, 79 and 71% of respondents, respectively. Among dish brush users specifically, N equals 1,402. The countertop is typically cleaned with a cloth, 51%, or sponge, 36%. 28% of all dish brush users use the brush to clean a chopping board after soilage from chicken juices, with highest prevalence at 38% in Hungary. It was not asked for other uses of brushes in the present study, but in a former investigation from Norway, 92% of respondents used brushes for hand washing of dishes, and they reported that they also used the brush for cleaning the vegetable drawer in the refrigerator, 25%. Trash bins 6%, and pet feeding bowls 2%. Jacobson and Lavik, 2011. Differences in the use of sponges and brushes may affect both the frequency and type of contamination, soiling and time for drying between use. Consumers show different motivations for changing their cleaning utensils, sponge or cloth. Relying on time, I do this at regular times. 65% of the households, on their senses looks dirty, 37% and when smelly or slimy, 22% and slash or on usage are for cleaning juices, 22% or dirt, 20% supplementary fig. S2. There were only small differences between countries not shown. We also ask for how long the respondents typically use the same sponge or cloth before changing to a new one. 29% of respondents change within 2 days, while 71% change after 3 days or more. Changing utensil every 2 days was more typically associated to usage wiping up juices, while changing less frequently was more typically associated to sensory motivations, look, smell or feel. Consumers reporting that they changed at regular times did not deviate from other consumers with regard to frequency of changing to a fresh utensil, supplementary fig. S2. The results corroborate earlier studies. In a survey conducted in Switzerland N equals 1,122, 52% changed the dish sponge every week or less often, 31% every 4-5 d/once a week, 17% daily/every 2-3 days. In an Italian study, all 100 consumers used a sponge for 5 days and longer, and 40% used it for more than 30 days. Looking into gender, age and risk group effects, results showed no notable gender differences in frequency for changing cleaning utensil. However, females more typically changed the utensil at regular times than males, 71% against 59%, while the latter relied on their senses more often than females, 39% against 35% for looks dirty. Further, with increasing age, consumers reported more usage of a dishwasher within equipped households and lower usage of a dish sponge, increase in change frequency of cleaning utensils and more change based on usage time and less based on sensory cues, supplementary fig. S3. Interestingly, about 3 to 5% more of the households including individuals at risk i.e., either pregnancy, young children, diabetes and immunodeficiency, or age over 65 years responded with higher prevalence of hygienic strategies compared to households without specific risks. A more frequent change to a clean or new utensil, decision to change more often based upon time routines or usage and less often based on senses, and more usage of a dishwasher not shown. 3. 2. Water uptake and drying of sponges and brushes. As shown in Fig. 2, there was a large variation in water uptake and drying rate among the cleaning utensils. The brushes only absorbed a low volume of water, car 5G, and were dry 0, 25G water after 4, 5H. Compared to the brushes, the sponges absorbed larger volumes of water, prox 8180G. The sponges dry slower, even if they were squeezed once after the initial water uptake before drying. The drying rate varied between the sponges and, as expected, large sponges dried slower than small. If not squeezed before storage, all sponges contained relatively high amounts of water, prox 4120G, even after 24H of storage. Squeezing removed 85% of the water, and after 4-5H of storage, the sponges contained 10G water. Residual water was only found in the largest sponge S22 after 24H1. 5G. Sponges may be used for several purposes. If considering wiping off surfaces like bench-ups, refrigerators, etc., there is probably an advantage that sponges absorb water, as this will ensure that the cleaning is wet, which is likely to be more effective than dry cleaning. Also, when wiping up wet spills, absorption is advantageous. Brushes does not take up water and are therefore not used for spills, but are mostly used for washing up under running water or on utensils immersed in water. The large differences in water uptake and drying rate among and between sponges and brushes may affect survival and growth of bacteria. 3. Growth and survival of bacteria in sponges and brushes. The growth and survival of Campylobacter, Salmonella and kitchen-associated bacteria were studied in 3 types of sponges and in 1 type of brush. The items tested were previously shown to have different water uptake and drying ability. Fig. 2 and were added water or soil daily except after 4 and 5 days of storage. There were differences in bacterial survival and growth between the type of cleaning utensil. The lowest bacterial numbers were found in brushes, where a rapid die-off of all types of bacteria was observed. Fig. 3. The rapid die-off corresponds to the fast drying of brush, as shown in Fig. 2, as no residual water was detected. 4. 5. HR for immersion in water. For sponges, rapid initial growth of bacteria was observed. Fig. 3. And the total bacterial numbers reached around 9 log after 1 to 3 days, and remained stable thereafter. These levels are in line with levels reported in used sponges collected from consumers. The levels of Salmonella and Campylobacter typically decreased over time, and the levels of pathogens in sponges were much lower, 0.000011%, than the total number of bacteria. Growth of Salmonella the first day after inoculation was found in sponge type S22, which according to the drying experiments dried slowly, and Salmonella was also detected after 7 days of storage. In average, S22 contained 5.6 g water after 24 h of storage measured each day during the experiment. In comparison, the two other sponges tested, S13 and S21, contained on average 1.3 and 2.8 g water, respectively. Thus, there are indications that Salmonella survived best in the most humid sponge. Campylobacter died off rapidly, especially in brushes. There was longer survival in sponges, where Campylobacter was found after 1 day of storage, but not after 3 and 7 days. From other studies it is known that Campylobacter is very sensitive to drying, compared to Salmonella, and this can explain lower survival of Campylobacter in dry brushes compared to more humid sponges, and also better survival of Salmonella than Campylobacter in sponges when those dry out over time. Both Campylobacter and Salmonella have been found in used sponges and cloths collected from consumers. 3. 4. Bacterial dynamics in sponges and brushes over time. The bacteriota analysis of the experiment with quantitative bacterial counts shown in Fig. 3 revealed that Enterobacteriaceae were dominant after 1 day of storage of sponges and through the whole storage period for brushes. Fig. 4. Further analysis separated Enterobacteriaceae in the two genera that were added as Serratia and Salmonella, and showed that Serratia was the dominating genus. One obvious explanation was that Serratia dominated in the inoculum, although the intention was to add all bacteria in equal numbers. The relative abundance of Serratia decreased from D1 to D7 in the sponges, but not in the brush. In sponge S21, Pseudomonas was completely dominant at D7, while in S13 Pseudomonas dominated together with Marsilia. In S22, Pseudomonas dominated together with genera typically found in the environment e.g. Chrysobacterium, Stenotrophomonas and Xanthanobacterium. An interesting observation is that these environmental bacteria together with Marsilia in S13 and P. Verona in S21 were not part of the bacterial mix added, and is likely to have originated from the added food soils, although the new cleaning utensils could not be ruled out as sources of bacteria. The increase in total bacterial count over time in sponges is likely due to high growth of Pseudomonas. Pseudomonas in general compete well at humid conditions but is sensitive to drying, and this could explain its higher prevalence in sponges than in brushes. The bacteriota analyses revealed that the pathogenic bacteria were only a small minority of all the bacteria present, confirming the results obtained using selective media for Salmonella and Campylobacter shown in Fig. 3. In initial experiments, performed in the same manner, it was revealed after the experiments were completed that Comamonas also from kitchen surface from Portugal mistakenly had been added instead of Serratia, due to a contamination of the inoculated strain. In these experiments, Comamonas was prevalent immediately after inoculation and after 1 day of storage, but it was found in very low numbers after 3 and 7 days, when Pseudomonas took over as the dominating genera. Supplementary Fig. S4. The Pseudomonas dominance was higher in these experiments than in the experiments where Serratia was added. Regarding the survival of Salmonella and Campylobacter, as revealed by plate count, the results were similar in the experiments with Comamonas and Serratia in the inoculum, a better survival of Salmonella than Campylobacter and highest survival of Salmonella in sponge 22. When interpreting bacteriota results, it must be considered that both living and dead bacteria can be detected, and that differences in copy numbers of 16's rDNA can affect relative abundances. In a German study, used sponges were found to be heavily colonized by Acintobacter, Moraxella and Chrysobacterium. In another German study, Acintobacter, Anhydrobacter, Agrobacterium, Pseudomonas and Chrysobacterium dominated. These bacteria were also found in the present study, although Agrobacterium was only found in very low number 0.15%. The model system used in the present study seemed to favor Pseudomonas and Serratia, but the reason for this is not known. It should be noted, however, that in the present study we tested new sponges in laboratory experiments, while the researchers in the German studies collected used sponges from consumers. 3. Viv. Antibacterial effect of sponges containing antimicrobial products. The sponges S21 and S22 were claimed by their manufacturers to contain antimicrobial products silver and quaternary ammonium compound disinfectant, respectively. In agar diffusion tests with bacteria spread on agar or embedded in soft agar, no growth inhibition zones were observed for S. Infantis, S. Enterotides, Pseudomonas sp. or Staphylococcus sp. Examples shown in supplementary Fig. S. Viv. Inhibition zones about 2 cm for all bacteria were observed for the absorbent, MAT containing zinc pyrithione positive control. In the experiment testing growth/survival of bacteria, the highest growth/survival of total bacteria and Salmonella were observed for the S22 sponge containing QAC Fig. 3. It is likely that high humidity and slow drying were more important parameters for determining growth and survival of bacteria than the content of QAC. For the S21 sponge containing silver, the growth of total bacteria was slower and the reduction of viable Salmonella and Campylobacter faster than in the other sponges tested. Fig. 3. However, it is difficult to conclude whether this was due to the presence of silver, as no inhibition was observed in the agar diffusion tests. It was also observed that S21 at day 7 had a different bacteriota than the other sponges, with absence of Enterobacteriaceae and being totally dominated by Pseudomonas veronii. To our knowledge, there are few other scientific studies examining antimicrobial effects of sponges' added antimicrobial compounds. Enriquez et al. 1997b reported that sponges containing an undisclosed antimicrobial compound had reduced number of total and fecal coliform bacteria in a dishwashing 5-day model study. In 2 studies where drops of antimicrobial dishwashing liquids active compounds not disclosed were added to conventional sponges, some of the liquids resulted in a 3-5 log reduced total bacterial count in sponges in one of the studies, but no effect on Salmonella and total bacterial count was observed in laboratory tests in the other study. In the latter study also no effect on total bacterial count in sponges was found in practical tests in kitchens where antibacterial dishwashing liquid was added daily to sponges. In general, for domestic products like cutting boards, countertops, sinks, etc., where antimicrobials are integrated/coated on the product, their effect on food hygiene are limited. Motru and Langsrud, 2011. Even if the added compound has antibacterial properties in vitro, when incorporated in a product there may be challenges like too low concentration, limited availability and declining concentration over time. These factors, as well as neutralization in presence of high organic loads, may limit the antibacterial effect under practical use. Motru and Langsrud, 2011. 3. 6. Cleaning of sponges and brushes. As described previously, used sponges and brushes may contain a high number of bacteria, and at least for sponges in some occasions also pathogens. To reduce the risk, sponges/brushes may be replaced by new items when contaminated. Cleaning of sponges and brushes may be an alternative to replacing them by new items. Observed cleaning methods during kitchen visits as well as other methods described in literature were tested in laboratory studies. After performing the cleaning routines, some sponges and the brushes were sampled immediately for bacterial enumeration and other sponges were sampled after 2 days further incubation after adding food soil or Salmonella. The purpose of adding the soil was to test if a low number of bacteria below the detection limit remained after the initial treatment, and for addition of Salmonella, the purpose was to test if remaining soil after cleaning treatment could support growth of bacteria including Salmonella. Based on the results for total bacteria Fig. 5, the cleaning methods tested could be ranked from the most to the least effective as follows: 4000 ppm chlorine dishwashing equals boiling washing in detergent water plus drying microwave oven equals washing in detergent water, no washing procedure. The ranking was the same for sponges and brushes and was supported by the results for salmonella plating on XLD. Data not shown. Chlorine eliminated bacteria to below the detection limit and inhibited regrowth. Dishwashing machine removed bacteria and most of the soil, but still some soil was left, allowing regrowth of bacteria. The cleaning effect may depend on the type of dishwasher and program. It remains to be tested whether economy programs with lower temperatures and water used are effective. New sponges and brushes, no treatment contained bacteria not salmonella that grew when nutrients were added. Cleaning in detergent water followed by drying removed/kill salmonella, but other bacteria remained after the cleaning. Introducing an overnight drying step after detergent cleaning increased the reduction of bacteria. Microwave treatment reduced salmonella and total bacterial count in sponges and brushes, but remaining soil and bacteria lead to regrowth. Boiling killed bacteria to below the detection limit, but remaining soil lead to regrowth of surviving bacteria or growth of salmonella when salmonella was added. Although not tested here, the procedure reported by some consumers with rinsing in water is not likely to be more effective than cleaning with detergent and water. The results obtained in the present study were mostly in line with the conclusions from other studies, with some exceptions. Chlorine treatment of sponges has also previously been reported to be effective, but in another study only low effect was observed on total bacterial count. This may be explained by possible quenching by food soils and low exposure times 3 min in the latter study. Sharma et al. 2009 reported high effect 7 log reduction on total bacteria by use of microwave oven for 1 min at 1300 W. In the present study we found only low effects of microwave treatment on the number of bacteria, maybe because the power level was lower 1 min at 700 W. The effect of different cleaning methods was similar for brushes as for sponges, with several effective methods. We have not found studies on cleaning of kitchen brushes in the literature. 3.7. Food safety impact of contaminated sponges and brushes. As salmonella and campylobacter may be found in sponges, it may be asked how large risk contaminated sponges are for consumers. The most likely scenarios for ingesting pathogens through the use of sponges are the transfer of pathogens from sponges to hands and then to mouth either directly or via food, or b from sponges to a surface/equipment and then further transfer to food. As only a fraction of bacteria will be transferred in each step, the direct route from sponge to mouth via hands will potentially transfer the highest number of pathogens. In eggs, concentrations of salmonella as high as 8-10 log CFU/ml have been reported. Clay and Board, 1991. Thus, in a worst-case scenario, when such spills are wiped up with a sponge, 10 log CFU salmonella may end up in the sponge. Assuming a 1% transfer ratio of bacteria from sponges to hands or surfaces, up to 6-8 log CFU salmonella may end up on hands or surfaces. In such a worst-case scenario, both direct transfer from hand to mouth or via food, as well as transfers from a utensil wiped with a sponge to food may lead to a risk of salmonella infection. But it should be noticed that such high levels of salmonella in eggs are not common. The maximum concentration of pathogens in raw poultry are lower than in eggs. Thus, for sponges used for poultry spills, only the direct route from sponge to hands to mouth, and not the route from sponge to equipment to food, is likely to cause a risk of infection, even in worst-case scenarios. If the food spill is allowed to dry on the surface before it is wiped off, the concentration and transfer of the pathogens, especially Campylobacter, will be even lower. When considering the risk of using contaminated sponges, it is likely less risky to use a sponge with your bare hands than touching raw poultry with your bare hands, as the concentration of pathogens are likely to be higher on raw poultry than in a sponge used to wipe up poultry spill, but it cannot be excluded that salmonella may grow to higher concentrations in sponges and lead to increased risk. The risk of ingestion of pathogens through the use of brushes is even lower than for sponges, as the levels of the pathogens will likely be lower in brushes due to die off because of drying. Also, since the brushes have a handle, this reduces the risk of contact with bare hands to the part of the brush which encounters contaminated food spills. 3.8. Advice from food safety authorities regarding cleaning utensils. When checking food safety information from health/food safety authorities in selected European countries as well as from WHO, few recommendations on the use of sponges or brushes were found, as most advice on cleaning utensils focus on cloths. Consumers were advised to wipe up meat spills with kitchen paper Denmark and France, or a disposable cloth Germany. There is generally a focus to clean and change clean utensils frequently. The use of sponges is not recommended in Germany or by the WHO. In Germany, cleaning brushes in the dishwashing machine is recommended, while in France cleaning/disinfection of sponges by boiling, soaking in diluted bleach or in a microwave oven are recommended. 4. Conclusions and advice to consumers. Advice for use of sponges or brushes from food safety authorities are limited. The present work supports the food safety recommendations for consumers from the WHO to not use sponges for cleaning. A good replacement for dishwashing is brushes. The use of brushes is already common in some countries, and adopting this practice in other countries should therefore be possible, though it would require efforts from the health authorities, educational institutions and market actors. In the present study, differences in cleaning practices between countries were found, variations in drying and survival of pathogenic bacteria in new sponges and brushes were observed, and cleaning methods were evaluated. These elements constitute the basis for the following recommendations: use brushes rather than sponges for dishwashing. It is more hygienic to use brushes when brushes dry faster, and there is a lower risk of growth/survival of Salmonella and Campylobacter in brushes than in sponges. 2. Since brushes have handles, bare hands will not be in contact with the water, allowing higher temperatures and thus better cleaning effect. 3. Hands will not be contaminated when using a brush, since pathogens will not be transferred from the brush to bare hands. When using sponges, this is a risk, for it is easy to keep brushes clean by using a dishwashing machine. Use paper or single-use wipes for raw food spills. Avoid using sponges in high-risk situations, such as wiping/cleaning up raw food spills. Using single-use wipes or paper may be alternatives in such situations. If sponges or brushes are used in risky situations, they should be cleaned/replaced directly after use, since the pathogens will likely not die off either time of next usage. Clean sponges and brushes with chlorine, dishwashing machine or boiling. The most effective methods for cleaning of sponges and brushes are soaking in chlorine 4000 ppm, 1620H, cleaning in dishwashing machine and boiling. It should be noted that the research in the present work was performed with new brushes and sponges, and we are currently investigating bacterial levels and the survival of Salmonella in brushes and sponges that have been used by consumers. Declaration of competing interest. The authors declare that they have no known competing financial interests or personal relationships that could have appeared to influence the work reported in this paper. Acknowledgements. This research was funded by the European Commission H. 2020. SFS 2016-2017, a project No. 727,580 Safe Consume. Suzanne Notchell and Susanna S. Rayton Lanks are thanked for providing bacterial strains. Tove Morgest and Merit R. Jensen and Seen M. Dromtorp at Nofema, and the technicians of the Synod Microbiology Laboratory Universidade Católica Portuguesa are thanked for excellent technical assistance. The authors wish to thank Joachim Skolderer for planning and managing the consumer survey.